Thanks everyone for coming today. So my goal today is to talk about how we might use some different types of tools to do a biodiversity change synthesis, or just even look for synthetic patterns, generalities with biodiversity data. So I'd like to start this talk with, what is meta-analysis? Because I'm going to build my way up and kind of try to make one of the points that I would like to make today is that meta-analysis is a fantastic thing, but it may not be the most useful way to synthesize biodiversity and variation in diversity. But we're going to start with the definition of what a meta-analysis is. And in English, whenever I see the word meta followed by some other word, I repeat the second word. And so the best way to think of a meta-analysis is an analysis of analyses. And it's one of the ways that statisticians and various other people concerned with drawing generalities from bodies of evidence have used quantitative tools to look for generalities and other aspects out of existing scientific evidence. And so it's this quantitative tool that has grown quickly, really in the last, what are we looking at there, since 1990, or certainly since 2000, in the last 25 years, we've had this really exponential growth of meta-analysis in general across the sciences. It was introduced from the medical sciences, okay? And so this paper from Jessica Gurevich and her colleagues in Nature, published a few years ago now, was, really kind of the history of meta-analysis, if you like, and how it's been used to synthesize evidence in the sciences. And so from the 1950s, we started to have some statistical tools being formalized. And the term itself was born, approximately 50 years ago in the 70s. It's now regarded as being one of the ways or a really important path for scientific progress, meta-analysis. And we can think of it as having two fundamental goals. And what I want to do to start this talk is to provide some evidence of case studies for these two fundamental goals that I've been lucky enough to be involved with. The first example that I want to touch on is that we can use meta-analysis really to assess the evidence for a specific intervention. And when we say intervention, it's, a manipulation, if you like, or an experiment in a lot of contexts. If we think of the medical context where meta-analysis was introduced, this intervention was often a medical intervention. So whether or not a treatment was being effective for different cohorts or different groups of a population. In my work, we've used the example that I want to give was led by a postdoc in the group that I'm in at iDiv named Emma Ladekasser. And she really wanted to know about whether or not a particular intervention, in this case, the addition of seeds, influenced diversity and productivity. Okay, so this is an example from grasslands where experimentalists were putting additional seeds, and when we look at a figure, we can think of the additional seeds being added as the number of species of seed added. So we can see on the x-axis here, the intervention is how many different types of species were added to a particular grassland. And on the y-axis is the response of species richness in those grasslands to that addition of new species. If you know anything, or if you're familiar with kind of the jargon associated with meta-community dynamics, when we add seeds to a meta-community, you can think of that as overcoming any kind of dispersal limitation. we're really now having all kinds of species, up to 60 species, now able to reach all of the patches in our meta community. And Emma was really interested in asking, how does that alleviation of dispersal limitation influence species richness? And she pulled together experimental data from what I recall was, I think, 12 experiments, and each of those experiments is represented by a different colored dot and different colored line on this graph. And we can see the overall average across the 12 experiments with the black line. And we can see that, on average, we got some increase in the number of species in our grassland communities per seed added. The more seeds we added, which we see on the right-hand side here, the more species we ended up counting in our meta community. And the second half of that question that Emma was interested in asking was, how did that impact the productivity of that community? And so various people refer to this as the biodiversity ecosystem functioning relationship, where the biomass and it really is the above ground mass of the plants that exist in the community, whether or not that responded to the species addition. And in contrast to the increase in species richness that we saw when we added species to the community, we really saw no change in biomass. And so this was an example of where we were using synthesis. It's strictly not a meta-analysis, and I'll come back to this point as we progress through the talk to try to make it concrete that here we were able to do a synthesis with some raw data. We had access to the species abundance distribution from all of these plots. We didn't simply collect effect sizes from the literature. And Emma was able to show that as we add more and more species of seed, we increase the species richness, but have no effect on biomass. As an example of how we use evidence synthesis and quantitative evidence synthesis to ask how this intervention of reducing dispersal limitation influenced the grassland community. The second way that we use meta-analysis, and this is certainly something that I've been involved with lots of projects, is to ask whether or not a particular pattern is general across many different aspects of the same system, or when we look across multiple systems. And I think the example that I'm going to give from memory is an example where we're looking across multiple systems. And this was something that the leader of our group led, Jonathan Chase, and he was interested in asking whether or not species in small habitat fragments were a random subset of those found in large habitat fragments. And to do this, he compiled lots of data, and there's a data paper associated with this where we published the open data, and he put together all of the studies and all of the data we could find where we had samples of different types of communities from different types or habitat fragments of different sizes. And so in order to be able to address this question, we really needed a sample from a small habitat fragment and a large habitat fragment. As we'll see in the data, quite often we had a gradient of habitat fragment sizes for which we were able to collect these data and put them together in order to address this question. This is just one of the figures from this paper to ask whether or not this pattern of the relationship between fragment size and standardized species richness was, increasing across many different systems. Each of the different colored lines here, I think from memory, represents a different taxa. You'll forgive me for not putting that on the plot. And you can see, the odd line actually shows a different relationship. Some of these lines look fairly flat. Some of them, I can see the edge of one that is even negative. But on average, we see this positive relationship between fragment size and species richness. And when we put that together with other metrics, numbers of individuals and evenness, or the relative abundance of the species in these communities, we were really able to find that in general, the species in small habitat fragments are not a random subset of those found in large habitat fragments. If that were the case, this line, and I don't go into the details, I'm sorry, we can talk about it afterwards. And I think I've got an example later on where I can illustrate what would happen if the number of species in small habitat fragments were a random sample of those in the large habitat fragments. We'd expect to see a flat line in this relationship. But here we can see that it's increasing. And so something else is going on in the small habitat fragments. John referred to this as ecosystem decay, and we can think of a number of different processes that might be associated with that ecosystem decay, mostly due to changes in demographic rates. We might imagine in small habitat fragments, mortality rates might be increased, or we might have changes to fecundity and things like that associated with living in a small habitat patch. With the data that we had together, we were also able to do some secondary analyses and ask whether there were any other patterns in the data. One of the patterns that emerged from our analyses was to do with the what is referred to as the matrix. And so this is the habitat in between, or it's in between the habitat fragments. It's how is the landscape? If it's a harsh landscape, we thought of that as a landscape that it was going to be hard for species to move in between the habitat fragments, as opposed to a light habitat or a light matrix where it might have been more easy to disperse between habitat fragments. And this plot this plot shows the slope. And so this is the slope of the relationship between habitat fragment size and species richness, how that responded to these different categories of the matrix. When it was easier to move through the matrix, we got shallower slopes. This line was flatter. The relationship between fragment size and species richness was flatter when it was easier to move between habitat fragments, and it got steeper the harder it was to move between habitat fragments. And this really suggests something about the processes that are going on. So if I am guessing correctly, I'm in an audience where you don't necessarily count things that you can see. And so biodiversity, I just wanted to step back a moment and think about what types of things we're trying to quantify when we talk about biodiversity. And there's some really general properties about biodiversity, whether we're counting things in a tropical forest like we can see here. And this is whilst I'm not really a terrestrial ecologist, I'm really most familiar with counting things that I can see, as opposed to some of you, I think, will be working with things that we cannot see. We might be thinking about the diversity of butterflies or plants. Our first example came from grassland communities. But I suspect for many of you, you're really counting molecular diversity, or something that I know to call OTUs. Is that going to come to me? Something taxonomic units. Biodiversity is this multi-dimensional thing that it really is a very broad term that can represent many, many different types of systems. But across all of these different systems, there are some commonalities. And the two commonalities that I want to emphasize today are that whatever you're counting, if it is biodiversity, it has these two facets or two things that are really important. One is that it's going to be a multi-component thing, and I'll detail exactly what I mean by multi-component. And also really critically, it's this scale-dependent phenomena. And I'll dive into both of these things in a little bit more detail. So when I talk about it being biodiversity being multi-component, the components that are most important are, first of all, we can think of the number of unique identities. For me, this is most often species richness, but for you guys, it may be these O's. What's the O? Clara, you might rescue me and remind me what the O is.
Continuous or operational taxonomic unit.
Operational, thank you. Maybe the second beer at dinner was too many. Operational taxonomic units could be the unique identities that are you guys are counting quite frequently. And then you have a number of reads. In my systems, we're most often counting individuals, ideally. And another really important component of diversity is how many of those different identities we counted, okay? So the total number of identities. And the final component that we're going to touch on is the relative abundance. Sometimes people refer to this as evenness, or whether we've got many common things or a few common things and many rare things is the much more frequently observed pattern. In the literature that I'm most familiar with, we refer to that as the species abundance distribution. And we really, in most systems, and I'm sure it's true of lots of the systems that you guys study, we typically find a few common things and many, many rare things. And I understand that in different systems where you're quantifying these microbiomes, we have more or less rarity. The way that I imagine some of you are familiar with visualizing these patterns is through rarefaction curves. I think for lots of you, you're more familiar with seeing number of reads on the x-axis, but in my world, I'm typically looking at numbers of individuals. And we can see here this non-linear scaling relationship. As we count more and more things, this total number of identities, on the x-axis, we accumulate more and more unique identities, okay? And so the y-axis is this first component, the number of unique things, and the x-axis is this total number of things that we counted. And with a bit of experience and practice, we can actually read a little bit of information about the relative abundance, the evenness of a given community by the curvature of this relationship. And so if we look at a comparison of two communities, we might see, for example, with the solid line, both of our samples here have the same number of things. I'm going to call them individuals, approximately the same number of things, but the different patterns of relative abundance in these two communities, the dashed line has many, many more common species than the solid line, results in different numbers of total richness, or the number of unique identities that we read on the y-axis. And so when we do our biodiversity accounting, we'd really like to be able to keep track of all of these things. And this means that when we look to do biodiversity synthesis, I've got biodiversity change in the title there, but this doesn't necessarily have to be change from time to time. It could be change from place to place, or even a comparison between any two things. When we want to draw together evidence from lots of different sources and look for these generalities, meta-analysis, this analysis of analyses where we collect effect sizes from the literature, can become quite limited straight away. And I want to give an example from a student's work that I was associated with, Elaine Holitzer, and it's no longer in preparation. I'm somewhat proud to say it's published in Ecosphere now, an ecology journal from the ESA, the Ecology Society of America. And Helene was interested in asking, are bigger protected areas better for protecting fish biodiversity? And the way she went about doing this is through a classic literature search. And so she went to Web of Science and put in some keywords that I haven't detailed here, and initially identified, over 1,712 studies that eventually she had to screen down. And one of the limits to doing this type of analysis for biodiversity is you're really limited to whatever metrics the studies used. And so we no longer have this ability to dive in and calculate numbers of species or numbers of identities, numbers of individuals, and the relative abundance. We're kind of limited to what we find in the literature. And Helene found 43 records that she was able to put together to begin this process of answering this question of whether or not bigger protected areas are better for protecting fish biodiversity. And this is the type of thing that she was able to do. She collected studies where they counted the numbers of species in a protected area of a given size and a fished area of a given size, and calculated something called the log response ratio. And you can see here, this is kind of the treatment over the control, if you like, where the treatment is protection. And so where we stop harvesting fish from a spatial area that we set aside from harvesting, and we do some calculation to calculate how big of an effect stopping fishing had on species richness. Helene was really asked that, so this is to emphasize that first limitation, that we can't just calculate all of the metrics that we are interested in. We can't address this multi-component nature of biodiversity. We're really stuck to whatever they calculated in the original studies. Helene put together all of these different dots represent samples from different studies, and along the x-axis we now have her main question, and that was, how does the change in size of the protected area, how is that related to the response of species richness to protection? And somewhat surprisingly to us, we really found this overall negative relationship between the response of species richness to protection and the size of the area that was set aside from harvesting or fishing pressure. Now, this surprised us a little bit, but there's a lot of good reasons why we might find this relationship. Saying strong things about causality is also another limitation of this type of meta-analysis. But I think the most likely reason for the smaller effect on species richness as we went out to very, very large marine protected areas is that it's much harder to enforce that protection from fishing in the large areas. And there's quite a bit of literature about that in the fisheries and related conservation biology literature. But the other point I want to make with this study is really asking this question, are larger protected areas indeed less effective? And I want to use this question here to really dig into the other aspect of biodiversity, which is its scale dependence. Okay. And so here I've got a little cartoon of what we were able to do with Helene's meta-analysis and why that may not be the whole answer to how to the comparison of a big protected area. This green shaded area is my protected area from fishing on the right, and we're going to compare that to our small area. In Helene's study, we're really only doing this local comparison of an inside versus out, okay? And so when we go to the protected area, we're really asking, on average, if we compare these one sample in versus one sample out, one sample in versus one sample out, on average, what's the change? But in a large protected area, we might expect different species to be found in different areas. And that is exactly what contributes to a scale-dependent phenomena of biodiversity. You guys have perhaps heard of something called beta diversity, and that really refers to compositional change as we go between different samples in this large area. And addressing that question of whether or not beta diversity changed was really beyond what Elena could do with a meta-analysis. Because similar to the way that we're limited to whatever metrics people have used in the literature, we're also limited to the scales at which they've quantified the response to protection. And so if they haven't quantified the response to protection across multiple samples inside a protected area, then we're just plumb out of luck, and there's nothing we can do about addressing this scale dependence. So the first limitation was we can't do the multi-component change when we do meta-analysis. And the second limitation is we can't do scale-dependent changes when we do meta-analysis. And really open data, which I'm sure Steph and Clara have been pushing you guys towards, can overcome both of these limitations. And so I want to give an example of something that I did for marine reserves where I was able to overcome this limitation of using effect sizes and show you what we can do when we have the raw data, so to speak. And so I want to step back a little step first to ask about and illustrate using this rarefaction curve how we might be able to visualize one particular hypothesis for how MPAs might affect biodiversity. So I didn't show in these slides, I'm sorry, the most common response to protection in marine protected areas is actually this x-axis. It's really common that when we stop fishing, we get more fish, okay? And the simplest way that just having more fish in protected areas might influence the species richness and the biodiversity of the fish community is through something called the more individuals hypothesis. And really, when we just have more species due to more individuals, rarefaction curves tend to sit, if not right on top of each other, like in my cartoon example here, but very close to each other, okay? And so here, I wanted to ask whether or not protection from fishing and the response of having more individuals meant that the protected area rarefaction curve essentially sat on top of the rarefaction curve in the fished area. And to do that, I needed to, instead of going to the literature and looking for effect sizes, I really needed to go to the literature and find the raw data, okay? And the other thing that I can do when I have the raw data is if I have multiple sites, so now here's my rarefaction curve in different sites, I can ask whether or not we have site-to-site variation. Do the rarefaction curves look the same when we do this comparison between the protected area in the green and the fished area in the blue? And if we start to see some site-to-site variation, we might get scale-dependent contributions. And the other scale so these are kind of site level or what's sometimes referred to as alpha diversity. And we can pool all of these sites together, pull them together to create some larger scale that is quite often called gamma diversity. And again, we can ask this same question: how are the components changing? Is protection just meaning more individuals and more species, but really no change in evenness, pulling this curve up and down as we go from the local scale to multiple sites combined together in the gamma scale? And the beta scale just really refers to what happens in between those two scales, the alpha and the gamma scale. The other thing that I was able to do with this work, because I had all of the raw data, was really asked whether or not the response of fishes differed between things that we like to eat on the left. So these are what I referred to as the exploited species. These are the target species of the fishery, and the things that we really don't like to eat, that if we catch them at all, they're really coming into our catches as bycatch, but it's really not something that we want to take to market. To address this, we compiled data from the Mediterranean. And so this is a map of the central section of the Mediterranean. I think this is the French coastline. This is an Italian island. And all of these green diamonds are protected sites, and the blue circles are fished sites from where we had data. You can see that I don't have a perfectly balanced design here. I have slightly more protected areas than fished areas. We have balanced sampling within each of these icons on the map. We have 3 transects. And in order to try to adjust for any other variation, I also gathered some additional environmental information, but I really don't want to get into that today. I really want to emphasize these components and the scale dependence of what we were able to do. So this is what the data look at the local scale, the alpha scale. And so again, I kind of said with some practice, we'd be able to read these rarefaction curves, and we can see from the title exactly what we found here, that protection tends to increase evenness and richness, okay? And so the evenness we can see because these green curves tend to sit above the blue curves, okay? And that pulling up of the rarefaction curves means that those communities are more even. And the other aspect that, or basically due to that increased evenness, we also have more species. We actually don't see a strong signal of protection influencing the numbers of individuals here. If protection was having a strong influence on the numbers of individuals, all of the green curves would stretch a long way to the right compared to the curves. But instead, we have the green curves getting pulled up and ending at higher points than the blue curves. So that's really an effect of protection on evenness and richness. These 2 plots, or perhaps we don't need to dig in too much because they're not the main focus today, but really this response to protection differed between the exploited fishes and the unexploited fishes. You can see here we begin to get a bit of a signal of increased abundance for these things that we target when we're fishing. And that's less true for the things that we don't target. These green curves on the left are being stretched longer than the blue curves. How these different components are pronounced, and also how that differs between different groups in our analysis. This is just to show the average across all of those different sites. And so here we can see more individuals. We've pulled the blue average to the right. We've got more individuals and slightly more species for the exploited species, where really we've got no change. These 2 points for the unexploited fishes are really very close together on the right. So what happens when we combine all of these curves together? So we can put them together when we have the raw data to begin this process of looking for whether or not we see scale-dependent effects. And the result that we find for these data is that we certainly see a strong effect across all of these reserves combined for the exploited fishes. We see more individuals. We've stretched this curve, the green curve compared to the blue curve. It's more even. You can see it increases more steeply from the origin here, and we've got more species. And so protection for these exploited species is really affecting all of the components at the gamma scale. And that's slightly different to what we saw at the alpha scale. Okay, and so again, with the raw data as opposed to meta-analysis, we can begin to look at all of how at all of the components and how they change across scales. Slightly different story for the exploited fishes. We can see that when we combine all of the reserves together, which is the far right point here, we've got very similar numbers of individuals and similar numbers of species. But the fact that these curves get pulled apart at these intermediate scales in the middle here also suggests that we do have some scale dependence, and that scale dependence is really influencing the relative abundance as we accumulate species across sites. Again, it takes a bit of practice to read these things off rarefaction curves, but I really encourage you to think about that. Think about looking at them carefully and beginning this sometimes somewhat painful process of learning how to read them. If you're really interested in saying detailed things about variation in biodiversity. So hopefully this is, well, so we can see with all fishes combined, so this is now putting both of these aspects of the community back together. We can see that we've got a little bit of an effect on total numbers of individuals, but it's mostly gone when we combine all of the species together. And most of the effect happens in the curvature here. And so at the scale of that little section of the Mediterranean where I've got my samples from, at that larger regional scale, we're really seeing an effect on the fish community due to changes in relative abundance. The individuals haven't changed that much, but the fish community in the protected areas has become more even, and that increased evenness means we get slightly more species in the protected areas than we do the fish. When we combine those two scales, we get the beta scale. And so we really used a framework introduced by a fellow called Robert Whitaker, where beta diversity is the ratio of gamma over alpha diversity. And this just shows the beta diversity for one part of that rarefaction curve. And it really suggests that for the exploited species, we have more even communities leading to this spatial variation in the species that we find in the protected areas, and that's true for both the exploited species and the unexploited species. But again, maybe we can discuss that if that becomes something that you guys are interested in. The key that I want to end this talk with, I don't know how I've gone for time, hopefully I've taken up enough time, is that as we go from meta-analysis to this idea of having open data, the questions that we can ask we can ask many more questions, okay? And so historically, or even today, we really see this a bit of a divide between empiricists, the people who are out there collecting data, and the people like me who are taking that data and trying to reuse it to ask questions. This divide between empiricists and synthesists exists because often people don't publish their data. We're seeing a bigger push from more and more journals that data should be made open in order to be published. But at the moment, that's not terribly widespread. I think you guys who are maybe working with molecular data actually have a longer history than people like me who are dealing with things that we can see, and things like GeneBank have been around for longer. But if I recall correctly from Steph, even GeneBank is a bit of a mess once you start looking for the data, and it actually can be hard to compile data to ask synthetic questions with data from GeneBank. Okay, but if we look to the future, maybe we can coexist more friendly with the people who are collecting the data and bring them into our questions and really make this interplay between the people who collect primary data and the people like me who take that primary data from multiple sources to look for generalities or whether or not specific interventions are working like we think they should. And this is kind of a paper from Shinichi Nakagawa, who's one of the leading quantitative synthesists in our field of ecology and evolution, for his vision of the future, where we can really work together with data collectors and synthesists to draw evidence, make these quantitative evidence syntheses together. So I think that is about all I've got for you guys today. These are some people who have kept me off the street for the last few years. I was with a lab at Tel Aviv University called the Marine Ecology and Biodiversity Lab, run by Jonathan Bellmaker. When I did the marine reserve work, I'm still at iDiv. They can't get rid of me. And most recently, I've been taking some money from the ERC.